everyone. Thank you so much. It's such an honor to be here. Thank you for inviting me. I'm Saee Paliwal. As Ma said, I'm a lead AI scientist at Benevolent, and today I'll be talking about healthcare applications of machine learning with a focus on drug discovery and mental health. And a bit about me, like Ma said, I have a background in physics and then did my PhD in a field called computational psychiatry, which sits at the intersection of machine learning and mental health, looking at to use the tools of machine learning to augment patient stratification and diagnostics. I started at Benevolent about 4 years ago and have been focusing on accelerating drug discovery using AI. So why AI? There are certain problems in the world that are more readily AI-able, which is to say, their objectives are clear, there are more concrete ways to clean the data, there's a lot of data, and variance is more easily tolerated. For example, image recognition or content recommendation both lend themselves well to incorporating machine learning or AI. Healthcare is one of those applications of AI that has the potential to yield the most social benefit, but there are real challenges to using machine learning methods in healthcare, specifically because we face the challenges that come with biology or behavior. For instance, you run into data problems like few reliable ontologies to help categorize a particular problem, sparse data that's contradictory, subjective self-reports or medical records, and importantly, difficulty evaluating what good looks like due to long lead times or noisy signals. Right now, I think the most productive role of AI in healthcare is to augment experts. Significant advancements can be made using human-in-the-loop AI systems. Two such applications that I'll talk about today, like I said, are drug discovery and mental health. So let's start with drug discovery. The structure of this part of the talk will be as follows. I'll introduce the problem of why accelerating drug discovery is necessary, or why it's important to use AI to do this. I'll discuss ways in which we at Benevolent construct biomedical knowledge graphs to start addressing certain needs. And then I'll talk about transductive methods that improve the throughput of drug discovery given this data. I'll then discuss the challenges of evaluating the quality of models given the complex nature of biomedical questions. So let's start with augmenting drug discovery with AI. Right now, the current model of drug discovery is failing patients. It's failing everybody, really. The processes are really high risk and really expensive. $160 billion a year are spent on R&D and drug discovery, $4 billion per drug, and 88% of new drug programs fail. And there's no such thing as fast failing, like in tech. Drugs take 12 years to go from hypothesis to market, and those that do make it to market are only effective for about 30 to 50% of patients. With over 9,000 diseases, with no treatments, hundreds of millions of people suffer without a cure, and importantly, healthcare is the fastest growing government expenditure to take care of people for whom the system is failing. There's a vast opportunity here to improve upon the status quo and de-risk drug discovery, R&D in drug discovery, and most importantly, deliver better medicines to patients who need them. The BenevolentAI platform is aimed at doing exactly this. At BAI, we combine data integration and AI to achieve what we call the 5 Rs, linking the right drug, targeting the right gene for the right mechanism in the right disease, all to the right patients. At Benevolent, we use natural language processing to extract relevant information from biomedical literature and combine this unstructured data with structured knowledge from databases to build an internal knowledge graph that captures biology in its connectivity. We then, using this biomedical knowledge graph, infer missing links in the knowledge graph in order to identify useful undiscovered drug targets that modulate disease biology. We then have a precision medicine team to identify possible disease subtypes and a chemistry team that helps optimize how we interrogate a particular drug target. Today, I'll be focused on the derived knowledge and target identification portion of our pipeline. So I'm going to start talking about how we construct our biomedical knowledge graph. I'll touch on some of the ML methods we use to extract information from literature and integrate structured and unstructured data to form this graph, basically. So our drug discovery platform is powered by a biomedical knowledge graph, which contains multiple types of data. As you see here, we ingest tens of millions of documents that yield hundreds of millions of edges. Alongside that, we ingest structured data yielding billions of omics data points, and we integrate commercially available patient-level data that yields billions of data points. In our graph, 36% of our relationships are AI-derived. And interestingly, the platform is agnostic to disease area. It's just a general representation of biology in connectivity, and therefore it's easily scaled up. So let's focus on the text-derived side of the data. Here you can see a standard example of information extraction, particularly named entity recognition. This class of NLP models focuses on identifying the plausible entity for every portion of text. For biomedical text, entities are things like protein families or genes or diseases, etc. Here we see that from this text, we've extracted that E-cadherin is downregulated in epithelial cancers. But now, in order to add this information to an existing edge or construct a new edge in a knowledge graph, the next step is entity linking, or grounding the entities that we identified in this previous step to an existing ontology of entities. And here's really where what I was saying earlier comes to play. The challenge that we see here is that there's a lack of standardized ontologies in biology. So it's really complicated, and it's hard to know in advance the variations that a certain name can have. You know, genes have multiple synonyms, diseases have multiple ways of being referred to, and mechanisms as well. This is a multi-class classification problem with each entity as a class, or could be a nearest neighbor problem where you embed the entities in some space and cluster accordingly. And this can be done in tandem with named entity recognition. So the process I talked about on the previous slide and entity grounding can be done together. You know, here we have gene DCTN4 as a modifier of this disease, and on the right is an example or a schematic of this new entity tagging plus entity linking joint model that we developed in-house that increases the accuracy of our entity tagging, even though ultimately it's a pretty noisy process. And for those of you who are interested, I encourage you to check out the paper that I linked here to learn more about this model. So now you've extracted all these relationships from the text, and you want to aggregate this information together, uniting and truing conflicting pieces of information. This is particularly important with biomedical literature, in which there's a lot of conflicting information. It can be the case, for instance, that 3 papers confirm a relationship between, say, a disease and a gene, and then 2 sets of experimental data actually refute this relationship. And this this is a distance supervision problem, as we categorize it, in which we can encode textual context for each entity pair and then aggregate them with pooling or attention. And then for each pair, we have one final representation of what we think is kind of the true relationship between those entities, whether it's positive in that an edge exists or negative in that there's no relation. So the final result, the result of entity identification and entity linking and edge aggregation, is a biomedical knowledge graph. The knowledge graph consists of edges where each edge has a head entity and a tail entity, and then a relation that combines them. For example, protein-protein interaction. In-house, our knowledge graph consists of several types of entities, like genes or diseases or pathways or compounds, and relationships span everything from biological association to kind of therapeutically linked. The final result is an undirected acyclic graph in which self-connections are possible. In this particular example, from a paper we published in 2020, we had a very chemistry-heavy graph, but the specifics of the composition of the graph depend on the question being asked. For those of you who are interested, this is the process that I described is described in more detail in this paper that I've linked here on this slide. So what's important to note is that the graph consists of positive edges, or things we think are true. And the challenge now is to predict those missing links and assess how likely these missing links are to be meaningful relationships, say, between a drug target and disease, maybe something we haven't discovered before. Which brings us to our next step, which is graph inference to generate drug target hypotheses for a disease. So this process is called target identification. When I say target, I mean gene or drug target. Target identification is particularly important in the drug discovery process because of the high rate of clinical trial failure based on lack of efficacy. Almost half of clinical trials, phase 1 and phase 2 clinical trials, fail because of efficacy failures. And efficacy here can mean a bunch of things, but the majority of these failures is that the gene we thought to be causal in the disease was incorrectly identified. So machine learning gives us the ability to train on the entire knowledge graph and predict missing links, particularly predict viable drug targets for a particular disease or mechanism. This method uses training on the biomedical knowledge graph uses multimodal data and a huge amount of it, such that we're able to provide inferences to accelerate drug discovery in a way that no individual human could do. No individual human can read tens of billions of or tens of millions of papers or look at hundreds of data sets. Of course, this kind of training results in a model that learns abstract features of biological plausibility with the ability to generalize over disease areas. And it's not entirely clear that this is the optimal approach. So, do you mind overfitting if you only really care about one disease? So here we've built a general inference engine that can generalize, again, across diseases. But this is the approach we've chosen to take in the hopes that this kind of regularization allows for novelty and will unearth connections that a human user will not see. So I'm now going to talk about model evaluation, which is a particularly difficult task in biomedical contexts. Oftentimes, model performance is measured on a single outcome, which can be incongruous with how model predictions are evaluated by experts and later tested in the lab. It's like this example with stopping pain. If you want to stop pain in a limb, you will succeed if you cut that limb off. But we agree that this is a terrible option. What we actually want to optimize for is to stop the pain, reduce overall suffering, and maybe things like cost or time. So there should always be multiple parameters to optimize. And machine learning models are usually measured against a single objective. And additionally, it assumes that the benchmark that you're training and evaluating against is representative, which is really hard in biology. So here's an example where it's very difficult to tell kind of which model is better or more appropriate. So say you're predicting drug targets for breast cancer. In comparison one, you see that model A, the predictions for breast cancer from model A are disjoint from the predictions for asthma from model A, whereas the predictions for breast cancer from model B are very similar to those predictions that model B produces for asthma. So it's interesting because here model A is distinguishing between breast cancer and asthma, which is kind of what you wanted to do, and model B is not. But then say that you see that model A produces predictions for breast cancer that are pretty distinct from predictions from thyroid cancer. And then you say, okay, well, is model A actually learning really unique features of breast cancers such that they're disjoint from thyroid cancer? Or is it actually that, it's just learning nonsense? Then it should be learning that those cancers are more similar. And say model B produces predictions that overlap with thyroid cancer like you expect. So again, it's really difficult to say that one model is definitely better than the other. Similarly, going back to the idea of generalization, which model is better here? Do you want a model for which performance increases given the increasing amount of prior information you have about a particular relationship? So do you want a model that confirms what you know to be true? Or do you want a model that is heavily regularized and actually performance decreases with increased prior information? So it's actually giving you information that's novel. And this is an extremely hard, unsolved problem in drug discovery. Here, I've kind of put some caveats of machine learning metrics for drug discovery applications into 3 tranches: into data, computation, and interpretation. I won't go through these in detail now. I will share my slides and encourage you to go through them yourself if you're interested. I think these are all outstanding questions in the field of drug discovery. And if you're interested in learning more, please check out our publications and some additional publications that are relevant that I've linked here. Okay, what I'm going to do now is pivot slightly and talk about AI for mental health. Particularly, I'm going to talk about the nascent field of computational psychiatry. Computational psychiatry is an interdisciplinary field that uses tools like computational modeling and neuroimaging to gain a mechanistic understanding of psychiatric disorders. What we aim to do is to construct what we call a computational assay, or a battery of quantitative markers that include things like behavior, self-report, and neuroimaging that we feel characterize a particular disorder. We then hope to apply this computational assay to individual patients to better understand how a disease manifests across a group of patients. Ideally, these computational tools would allow us to identify subtypes within a particular mental health disorder, which is sorely needed given the heterogeneous manifestation of given mental health disorders across patient populations. The dream is then to identify patient subtypes and link them to particular treatment options, which are tailored to that particular patient group, and increase the probability that certain medication will be efficacious. This is where I did my PhD. I did my PhD at the Translation Neuromodeling Unit at the ETH in Zurich in this field of computational psychiatry. So I'll present results from one of the studies that we did. But first, I'll talk more generally about the tools of computational psychiatry. So in order to construct a multifaceted computational assay, computational psychiatry rests on 4 main tools. Naturalistic game-like paradigms, which we just heard about in the previous talk, modeling, neuroimaging, and questionnaires. So naturalistic paradigms are basically just games. You want to create a game that's really engaging for the user, such that the user exhibits the behavior that you're interested in modeling. These are passive measures of human behavior. These paradigms essentially produce an output upon which you run your computational models, in addition to neuroimaging. The machine learning side of this domain is primarily generative models that are inverted on human behavior and neuroimaging to estimate a set of parameters that characterize the computational mechanism involved in how this behavior was generated. There are many ways to go about modeling human behavior, but the generative approach, in my mind, starts to help to get us to a mechanistic understanding of what's going on. Combine this with neuroimaging, and you're able to put together this computational assay that helps us mechanistically diagnose patients. And finally, with mental health, you're unfortunately reliant on self-report, even though there are confounds here. And so since this field is still in its inception, what we generally do is we validate our computational approach using questionnaires or self-report. So the questionnaires become kind of the dependent variables in our workflow. What I'm going to do today is give you a quick example of what this process in understanding the development of impulse control disorders in Parkinson's disease after deep brain stimulation, or DBS, to the subthalamic nucleus. About 6% of patients undergoing DBS, Parkinson's patients, develop an impulse control disorder, or an ICD, after this operation. These ICDs include pathological gambling, or hypersexuality, or compulsive eating, and they can be really detrimental to the quality of life of the patient. Our goal in this study was to see, is it possible to use the tools of computational psychiatry to better understand the behavioral characteristics of an ICD, with the ultimate goal of predicting which patients might develop an ICD postoperatively, such that their treatment postoperatively could be tailored accordingly. I've linked the paper here for those of you who are interested in learning more. So, like I said, in this study, we used a naturalistic slot machine gambling paradigm that was really multifaceted, but essentially we created a slot machine for which the reward schema was variable. There were periods of high reward and periods of low reward, and we looked at essentially what bet sizes the gambler placed, or in this case, the patient placed while playing this machine. Data from this game was then used as the input to a hierarchical uncertainty encoding model. The model that we used was called the hierarchical Gaussian filter. This model involves a series of coupled Gaussian random walks, shown here as the x2 level and the x3 level. The evolution of these variables, x2 and x3, is governed by the step size of these random walks, which is dictated by a few uncertainty parameters, namely omega and theta that you see here. For our purposes, the most important thing is that these parameters, omega and theta, represent kind of the uncertainty encoding that the subject has when viewing the particular stimulus or making a particular decision, because we have a hypothesis that uncertainty encoding relates to impulsivity. So what we do is we have a patient play through the slot machine. We invert this model based on the observational variable u. And when I say invert, we basically estimate parameters omega, kappa, and theta. And then we kind of see if a subject-specific omega, kappa, and theta relate to any other variables that we have observed of the patients or that the patients self-report to us. In our study, we had 38 subjects playing the slot machine before and after deep brain stimulation. What we see, as we kind of expect, is that bet size increases significantly post-DBS. What's also interesting is that self-reported impulsivity, the Barrett impulsive, as measured by the Barrett impulsiveness scale, which is just a questionnaire asking people about their impulsive tendencies, significantly increases post-operatively on a group level. And similarly, what's really interesting is that we see an increase in parameters omega and theta. And remember, like I said, omega and theta are representations of uncertainty encoding. And so we kind of see that uncertainty encoding increases, or uncertainty increases post-operatively. And this is on the group level. And then On the individual level, kind of to lend construct validity to this approach, we look at the relationship between self-reported impulsivity via the Barrett Impulsiveness Scale, the BIS, and our model parameters. And we regress out to depression using the BDI, or the Beck's Depression Inventory, which is another questionnaire, which is a pretty widely accepted method. And what we find is that the BIS relates significantly to model parameters, both pre and post-operatively. But most importantly, the BIS relates significantly to omega post-operatively. So we can interpret that to mean that uncertainty around the probability of win in the slot machine relates significantly to self-reports of impulsivity. And again, this is just a proof of concept that basically the parameters that we estimate during live gameplay relate to self-reported characteristics of impulsivity from the patient. It's a proof of concept that we can actually measure impulsivity characteristics passively through an engaging game-like task, instead of having this person answer a questionnaire every day. You know, questionnaires are really subjective self-reports, so being able to passively measure this characteristic is useful because you can have someone play a game every day or several times over the course of a month or several months of treatment, for instance. You can gather a lot more data and kind of see how this behavior evolves over time. This could have, significant applications to early identification of behavioral addictions. If you see certain characteristics in the time course of impulsivity, you could predict that will be an onset of an impulsive incident. And I'm really curious to see how VR for mental health evolves, because VR provides you this really potent environment to elicit the behaviors that you're interested in. And so it might be a really great way to collect this behavioral data. I'm going to take my last minute, sorry, to talk about something that's really dear to my heart, which is data diversity, and something that we're working on at Benevolent. So one of the most important considerations, this was kind of touched on in both the previous talks, for AI and ML for medical applications, is the diversity of the underlying data. The data might be clean and be verified and be pre-processed, but if you dive into the makeup of the patient cohorts used in clinical trials or the patient cells used in phenotypic assays, what we see is that 81% of participants used in GWAS, for instance, are of European descent. Or another case found that 40% of clinical trials exclude the elderly. Or alarmingly, 1/5 of new drugs approved between 2008 and 2013 showed racial differences in drug response. And anywhere from 22 to 42% of studies exclude the sex of the animal subjects. There is a great need to increase the diversity of patient cohorts. As we're seeing with COVID, Black and brown patients are disproportionately affected, and we need to better understand the biological foundations of why that might be. At Benevolent, we started to look at our responsibilities in this domain, and we're thinking about ways to be part of the solution and increase awareness across the industry. This is a really incredible opportunity for AI and ML to make a difference in our ability to help patients. I encourage you to check out our panel on our data diversity initiative. And if you're interested in learning more, here are some resources. And overall, at Benevolent, we are hiring. So if you're interested in working in this really exciting field and contribute to things like the data diversity initiative, please get in touch. I'm really excited to get to know you better. Thank you so much.